Hello? Is this working? Can you see me? Do you know why you can see me? Because of your eyes, of course. Today we're going to learn about this amazing system, the optical system and the eye. And one of the best ways to learn about systems is to take them apart and see what they're made of. So that's right, kids. We're going to learn about the eye through a dissection. Now, in your notes, you're going to see a diagram that looks something like this. I'm going to explain to you what all of these different parts are and give you definitions while I'm doing the dissection. Let's get started. Now, we are using preserved eyes, meaning they have been soaked in a chemical that keeps them from rotting. This will cause the living tissue to change over time, and I will try and illustrate the differences in our dissection. So here's our sheep eye. First, we need to remove the extraneous tissue, the eyelids and the other extra skin and fat from around the eye. Now that we're done clearing the excess tissue, we want to make our first incision between the sclera and the cornea. The sclera is typically the white part of your eye surrounding the iris, and it provides protection and form for the eye. To make this incision, we'll use our scalpel to press into the flesh and point the eye down because there will be some liquid freed when the incision is made, as you can see here. The liquid that we freed is the aqueous humour, and it lies between the cornea and the lens. It's a fluid that nourishes the eye and helps to maintain pressure. Now that the first incision has been made, we can use scissors to remove the cornea from the eye. The cornea is on the outside of the eye and it's transparent, providing protection for the eye and also responsible for focusing light. So this is the cornea. It is typically clear in fresh living tissue, but because this eye was in preservative, it has become quite cloudy. Once we've removed the cloudy cornea, we're able to see the iris more clearly. The iris is the colored part of our eye, but it's composed of muscles to open or close the pupil. It controls how much light enters the eye. The hole that you see in the center of the iris is actually the pupil, but more on that later. First, we're going to remove the iris by making an incision and cutting around the entire muscle. Now, in order to better see the rest of the components of the eye, we need to remove the iris, and it also lets us see the underside of the iris better. The muscles that make the underside of the iris look almost like the ribs of a mushroom, but they're really two different kinds of muscle that control the size of the pupil. In order to show you the lens, ciliary muscles, and vitreous humor, I needed to make some small incisions around the opening so it can be more flexible. Then I'm going to flip the eye over and try to squeeze out what's inside. Sometimes it works better than others. Today was only okay. What I want you to see is that the lens is very cloudy, just like the cornea was. This is again because of the preservative. Normally it would be clear and quite flexible. It's attached to the ciliary muscles all around it, and the vitreous humor is the jelly-like liquid that fills the eye and helps it keep its shape. Now the lens is flexible because it is responsible for focusing the light entering the eye onto the retina at the back. And sometimes that means that the ciliary muscles, the muscles surrounding it, have to stretch it out or squish it together to make it bend the light in just the right way. We'll learn more about that later. And of course, as I said, the vitreous humor is the fluid that helps the eye keep its shape. Finally, the light is being focused onto the retina. This receives the light and sends signals to the brain via the optic nerve. Now, in order to show you the retina, I need to flip the eye inside out and detach it from the back. Now, you will notice on the sheep eye this cool little area that makes the retina look almost iridescent. This is called the tapetum. It helps some animals see better in lower light, and it's why when you shine a light on an animal, they appear to have glowing eyes. It's the light reflected off that part of the retina. So here we have the retina being detached from the back of the eye. This paper-thin piece of dark tissue is what's responsible for us seeing everything the way we do. This is where all of the receptor cells, the rods and the cones are. And there we have it, a basic look at a mammalian eye through a dissection.